All right, welcome everyone to the session on translational aspects. Dr. Laura Hunter was not available to facilitate this session, so I'll be standing in on her behalf. And to begin the session, we have Ludmila Prokunina Olsen from the Division of Cancer Epidemiology and Genetics at NCI. And she will be speaking on mutational signatures and translation, integrating germline and somatic genetics to explore cancer susceptibility, etiology, and outcomes. Mila?
Okay, can you see my slide?
Yes.
I assume, okay. So I'm very happy to participate in this very exciting and important discussion. And I will be talking about germline susceptibility, which is my favorite topic, and also it's a gateway how we got into this mutational signatures business. So we started from genome-wide associations of bladder cancer susceptibility, and this has been published in 2010 initially, where we identified that one of the device signals is a marker, or actually 3 markers on chromosome 22. And this chromosome encodes 3 APABEC genes. And all of them you may recognize as important enzymes in APABEC mutagenesis. And you can also note that this variant, which was associated with both bladder cancer, and then I will show with APABEC, is very common. It's between 35 to 66% in different human populations around the world. So as I mentioned, we first discovered it in 2010, but at that time, you guys did not publish your groundbreaking papers yet, so we didn't really have a lot of things to think about how it can be related to bladder cancer. And it took 2-3 years before all of you who, so everyone who produces papers are now in the audience, and thank you very much because you gave us a lot of material to work with, and that's what we did. So in addition to identifying this UAS marker associated with bladder cancer as a typical EQTL, so expression quantitative trait locus associated with expression specifically happen to be of A3b of a specific isoform. We found that the same SNP was associated with the count of APABEC signature mutations in the same direction as association with mRNA expression. And it was done in TCGA muscle-invasive bladder tumors, and this will come important that we are really going to be differentiating muscle-invasive and non-muscle invasive bladder tumors. So this has been already brought earlier today, that it was kind of counterintuitive, but we observed that both the risk for bladder cancer and then also mutation counts, mutation loads in these tumors were associated with actually better survival for muscle-invasive bladder cancer. And that's an interesting point of discussion. So based on that, we proposed that germline SNP or environmental factors such as viral infection or DNA damage would induce expression of enzymes. And I agree that we should talk about these 2 enzymes together. It's really hard to separate their function in this complex environment. And this will result in APABEC activity and APABEC mutagenesis, and will lead to increased bladder cancer risk, but a stronger immune response and better survival in this muscle-invasive bladder cancer. And we also established that A3b is predominantly induced by DNA damage, and A3a as a typical interferon-stimulated gene, which is induced by viral infection and other exposures induced by interferon response, including in immune cells. And there's another common variant within the same region, which is a deletion of 30kb, which eliminates A3b and creates this chimera between A3a and A3b, which actually encodes A3a enzyme. We showed that it's not important for bladder cancer, but it's a It's likely to be important for breast and other cancers. And it's also very common in some populations, especially in Asians. And overall, this is a distribution in populations. And then pan-cancer analysis showed that this SNP, which is tagging the deletion, the only SNP which is tagging the deletion, is associated with increased A3, decreased A3B type mutagenesis and decreased A3A type mutagenesis. And we proposed the hypothesis that this risk of genotoxic effect of mutagenesis exists already in normal tissues because of the different expression of this A3B enzyme, but nothing really happens yet because you did not probably reach this genotoxic threshold. And then with additional insults, this accumulates and accelerate the tumor initiation and development, which is additionally contributed by cancer therapies. And then this outcome is really going to be a combination of several factors. And based on neantigens and immune surveillance and synthetic lethality, high loads could be good. But again, in some circumstances, based on some specific treatments. And but still, it could be also counterbalanced by other factors. So this represented common variations, but there are, of course, uncommon and rare genetic variations, including those which already been mentioned here today, leading to microsatellite instability and different loss of mismatch repair genes and Pol E, Pol D mutations and nucleotide excision repair. So it's all important because they can produce maybe not extremely common individually, but in combination very common mutational signatures. But there also have to be some insults to initiate this process where this action of these genes will be required, such as radiation, UV, chemicals, and so on. And this is some interesting website which came from a recent publication in Nature Genetics for mining this kind of integration between somatic and germline alterations. So this is interesting to look because it has a huge number of patients in cancer types. Okay, I'll switch gears now and talk about that mutational signatures actually consist of individual mutations that can be very informative for various analyses, and also that as any crime, mutagenesis requires means, opportunities, and motives, and in our case, it's actually motifs. So we are working on bladder cancer. We know that this specific gene, FGFR3, is very interesting because it is mutated in 65% of non-muscle invasive and only 15% of muscle invasive bladder cancer. And among all mutations in FGFR3, which can be detected in at least 2 individuals, this mutation is the most common, which is present in 63% of all recurring mutations. So the question is, why? What causes this mutation and why is it so common? Looking at this motif of this mutation, we looked at actually motif of this mutation. We saw that it most resembles the signature 13 sequence, but not exactly. So we had a question: would it be consistent with the APOBEC mutagenesis? And there are means, because we determined that this mutation requires APOBEC expression by bioinformatic analysis in different data sets, and also by in vitro deamination assays, which is testing specifically APOBEC expression. In this case, it was APOBEC3A, and we can clearly see that there is a deamination reaction for this specific sequence, but not other sequences. And there is also opportunities because this mutation occurs more often in ligand strand of replicating templates. And also this interesting hairpin, which is formed by a sequence, and the mutation is located in the loop of this hairpin, which makes it extremely accessible and located in a single-stranded DNA, which makes it also a very good target for being accessed by APOBEC. And then, of course, motifs. It's being located in this sequence, which is often even overlooked as a typical APABEC site, but we showed that it's actually well recognized, at least in vitro, by APABEC activity, and we think that it's really an APABEC type motif. So, which brings the question, if this mutation is a perfect crime of opportunities, and maybe individual components not maybe completely optimal, but in combination, it creates this perfect combination when even the less optimal motif can be good. So why is it important to know if this mutation is APABEC type or not? Because knowing that, we can search for possible causes and also see if these factors can be prevented or mitigated. And coming back about muscle invasive versus not muscle invasive cancer. So this non-muscle invasive cancer where FGFR3 mutation is very common, is actually the lowest stage and low grade type of bladder cancer, where we don't expect this will be contributed yet by any treatment, especially by some chemotherapies like cisplatin and so on. So, and we don't expect also that there will be so much of genomic instability that it would be feeding this endogenously, this excess of APABEC mutagenesis. So where is it coming from then? And it's such high frequencies. It could be one common or several less common etiological factors resulting in same effect. And thinking about these factors, of course, we thought about tobacco smoking because it's the most common factor in bladder cancer. And to explore that, we analyzed about 2,000 bladder cancer patients with data for these mutations and other mutations, age, sex, and well-defined smoking status. And we specifically looked at this mutation, which we assigned as APABEC type. And then we also looked at this Y375C mutation, which is second most common mutation, and is RCC2 type, which is due to defective nucleotide excision repair caused by tobacco carcinogens, or as it's been linked to. And we also looked at some TK3CA mutations. So to make a long story short, and it's also been presented by Nina Rao in her poster. So we saw that this Y375C mutation is significantly more common in smokers as we would expect, but S249C is significantly more common in never smokers, which means that APABEC mutagenesis is not caused by smoking, at least in this situation, in this cancer. Okay, if not smoking, what then? What else could cause such a mutation? And we thought about viral infection, especially because of BK polyomavirus has been already linked to infection in epithelial cells of human urinary tract, such as bladder and kidney, and it's detectable in low levels in urine. And by adulthood, basically everyone is infected, but it's in latent state. And reactivation happens in immunocompromised conditions, such as in organ transplants. So we looked at this data sets, at available data sets by RNA sequencing, and in general, we don't see a lot of BK virus, but it's been detected in about 5% of non-muscle invasive and only one muscle invasive tumor in TCGA. And in general, BK is the most common virus detectable in bladder tumors. And we also confirmed by LTAG, by IHC staining. And in conclusion that, okay, so it does not really explain everything, and we didn't really expect that, but it explains some of the contribution to APABEC activity. And also, it was interesting to see that we can detect BK not in a just organ transplant population, but a general population of bladder cancer patients. And these tumors with BK are enriched in these specific mutations in APABEC type and FGFR3, and actually TK3CA as well. And of course, this does not answer all the questions, which means that we have to look at some other exposures, environmental, occupational, And we are doing this work with our colleagues from NCI, such as Stella Koutros and other collaborators. And I will switch to the last topic about that APABEC mutagenesis is not just a biomarker, but it's also a possible target for modulation. By doing our genomic work, we observed that there's alternative splicing in basically in many genes, but for APABEX3B is an interesting alternative splicing because it involves exon 5, which is really can be excluded and included. So when it's included, it creates this A3B1 as the designated form, which is mutagenic. And when it's excluded, it creates A3B3 form, which also we just designated. It's not an official name, but it's just a working name, which is non-mutagenic. And on top of that is actually extremely well eliminated by non-cis-mediated decay. So you wouldn't even see it much because it's constantly eliminated from RNA population. So the idea was that if we shift splicing from A3B1 to A3B3, would it hopefully decrease APABEC mutagenesis? So we did that, and we also observed that there's a lot of potential vulnerability in this splicing, specifically because the inclusion-exclusion of this exon is regulated by the spliceosome complex. And Ralph, who was the lead on this paper, he noticed that here are the papers that this pladienolide compound, drug, and some derivatives of that, they can be already used. They are used for clinical trials for cancers and conditions which required sensitivity of mutations in spliceosome factors. So we decided to try this drug in our system, and indeed by modulating how much of this exon 5 could be included or excluded, we could modulate how much of one or another transcript we can produce, and also then how much of protein we can produce, and then how much of deamination activity it will result with. So to make the long story short, that we actually can modulate APABEC mutagenesis by modulating activity of APABEX3 enzyme, and also by shifting the inclusion of this exon. And this last plot I highlighted because it shows that we're actually showing that this is working even when we induce APABEX3B1 expression by bleomycin, which is a chemotherapy agent. It's not cisplatin, but it's in the same line of inducers, and we know we use it as a positive control. And even when we do induce APABEX first, and then we are still able to decrease APABEC protein expression by pladienolide. And this also shows that digoxin inhibits non-cis-mediated decay and prevents elimination of A3B3, and that's why we can actually see that it's confirmation that it's a way to eliminate this non-functional protein. So we also were able to show that we can achieve the same effect by using a single splice switching oligos to also modulate inclusion. So in conclusion, we show that yes, APABEC mutagenesis have been associated with chemotherapy resistance, clonal evolution, tumor progression, and this all suggests that if we would do something to APABEC mutagenesis to modulate it, it might be beneficial in some specific conditions, and maybe transiently. So maybe just before chemotherapy, or whatever conditions could be relevant in this situation to prevent all of that chemotherapy resistance and evolution and so on. And so for now, we're showing that we can do it by addressing the splicing of exon 5, which is the most vulnerable point. And overall, I showed you today that there are multiple levels of regulation of APABEC mutagenesis. on the level of germline variants, environmental exposure factors and infections, and also alternative splicing. And this results also through additional modifying factors, which I didn't mention, for example, U and G factors, which are actually defining how much of mutations will be produced after this damages caused by APIBEX. So then we can look at this mutagenesis both at the whole signature level or at the level of specific mutations, and then we can connect to clinical outcomes. And these are translational opportunities which are on multiple levels for modulating levels and then for applying these biomarkers to clinical outcomes. And I would like to thank multiple people and collaborators from other places, and specifically to Ralph, my still research fellow, but he's starting his faculty position in a couple of months, and Nina Rao, who's a PhD student, still finishing this project. Thank you.
Thank you, Mila. That's a very, very nice presentation. Want to remind attendees that you can submit questions in the Q&A box, and panelists can ask their questions through the chat box. I'll start with a question. And so these, I'm interested in the variants that you talked about, and it looked like in different populations, the relative frequency of that variant was varied. So are those in populations, are those variants under selection? I guess is the first question, like a positive selection. And if so, I'm wondering what sort of forces are putting it under selection. For instance, if it's APOBEC, maybe it's viral infections. I just wondered if you had thoughts there.
Yeah, we have thoughts. We don't have a lot of proof, but I know that some other papers have been showing that some of these variants could be under selection, although selection would not be very strong because it's a minor factor for infections that would kill you. And there's definitely some variability in populations which are not really easily be explainable, but it tracks with the incidence, for example, of bladder cancer, which is most common in European populations, and the A3B SNP is most common in Europeans, up to 66% in Europeans. And this SNP is only associated with bladder cancer, no other cancer. So, you know, it's a lot of gene-specific, tissue-specific regulation for a specific SNP, not two SNPs away, but this specific SNP is regulating A3B expression, which is relevant for bladder cancer.
Okay, thank you. We have a question from Ruben Harris. Is the FGFR3 site edited at the RNA level or only at the DNA level?
It's a good question. I cannot tell you from the top of my head because actually the mutations for red, no, I can. So it's because it was actually compared both on DNA level and RNA level, but I know that actually the mutations in some cases were read from RNA sequencing data, but in many cases it was also confirmed by DNA level, by DNA sequencing or DNA genotyping.
Okay.
It does not exclude that it's not edited.
Okay, very good. Well, I think it's time to move to our next speaker. So thanks again, Mila.
Thank you.
And we have next Nuria Lopez Bigas, who's at the Institute for Research in Biomedicine in Barcelona, Spain. And her title is Mutational Footprints of Cancer Therapies. Nuria?
Okay, thanks for the introduction. You see my slides and you hear me, right?
Correct.
Okay. Okay, so I'll talk about mutational footprints of cancer therapies, and the reason we thought initially to study that is because many chemotherapies, not all of them, but many of them damage DNA one way or another. Here I just show you the example of some alkylating agents that produce some DNA modifications, which can be repaired by several repair pathways, but cells that don't die and the repair and the damage that is not repaired, when the cells replicate, they may lead to mutations. And then we thought that it would be nice to see if we can identify these mutations directly in human samples. One of the reasons why we thought of that is because chemotherapies cause long-term and late side effects, such as leukemias, for example, or cardiovascular morbidity, and a number of different late effects that maybe some of them may be related to the accumulation of mutations in cells in the body. But the first thing is that seeing the chemotherapy mutations is not easy, because if we see this cell that has already some pre-treatment mutations, at the time of treatment, there will be DNA lesions, and a cell that doesn't die, that survives and replicates, may convert some of these lesions into mutations. But seeing these mutations is not straightforward if we do bulk sequencing, because these mutations are going to be private at the time of occurring. They are going to be different in different cells, and they are going to have a very low variant allele frequency, so they are undetectable by bulk sequencing, while the pre-treatment mutations, if this is a tumor, for example, there would be clonal mutations. But if we wait for some time and there are some selective constraints and bottlenecks, some of these mutations of the chemotherapy may get into a higher variant allele frequency, so that we can identify those. And thinking on that, we thought that metastases from patients that have been treated provide a window of opportunity to find these footprints of cancer therapies. And the reason is because metastases, you don't have access to many tissues of patients treated, but metastases you have it because you have clinical biopsies. And also because there is a clonal expansion. During the chemotherapy treatment, there is a clonal expansion, and basically when you do a metastasis, there is a clonal expansion and a bottleneck that may give us this opportunity. The work I'll present now is by Oriol Pich, Ferran Muiños, and Anabel González Pérez in the lab. I think the timer is not on. I can continue, but just to let you know. Okay, so and the opportunity came because of this Hardwick data that we managed to get access to. I'm sorry that I didn't know that people in the US cannot get access to this data. For us, we applied for it and we obtained access. And at that time that we did the analysis, there was 3,500 metastatic samples with whole genome sequencing, which is what we need to identify mutational signatures. The other thing that we needed is very well annotated data, in particular of the treatment that this patient received. And that was also provided. And for each of these patients, we know if they received a platinum-based drug or some other type of treatment. So the first thing we did is to extract mutational signatures with two methods, Seq Profiler and Signature Analyzer. And then we search for mutational signatures that are associated to the treatment. Very simply, we were searching for mutational signatures that were very highly exposed or with high contributions in patients that are treated with X, and very low or none in patients untreated with X. This is just a cartoon to show you what we are searching for. The analysis has many challenges, in particular that we have many different tumor types, and as you know, the signatures also correlate with the tumor type. Also that the treatments usually are combinations of treatments, so we may have several chemotherapies or chemotherapies and targeted therapies given together to the same patient. And we had to solve these hurdles to be able to identify these associations. I'm not going to go into the details because this is published, but I'm just going to say that we did this analysis and we gave the opportunity to all the signatures and all the treatments to see if they are associated. We didn't want to limit by the ones that we know that can create DNA damage, but we did it in an open way. And as expected, many of the associations don't lead to any significance, but these are the interesting ones that are mutational signatures associated to some treatments. Here you can see some of them. Those are for platinum-based drugs, and this one for capecitabine or 5-fluorouracil. I'll just show you a couple of those. This is the cisplatin mutational signature that we pull out from this data in the metastases, and it's very similar to previously reported one. We also identified this one for capecitabine, which is a drug that metabolizes into 5-fluorouracil, and this is an analog of thymine and also blocks the synthesis of thymine. So it changes the pool of nucleotides, and it leads to very strong T2G mutations. Probably many people in the audience recognize this as very, very similar to signature 17 that it was shown before, which is common, for example, in esophagus cancer, and we were also puzzled by this. And then we wanted to validate that this is really due to the 5-fluorouracil. And one way that we found to validate this is by analyzing some data that was published from Leishmania, treated with 5-fluorouracil. The purpose of this work was to identify resistant mechanisms, because 5-fluorouracil is also used for leishmaniosis. And what they did is at the end sequence the whole genome sequencing of this Leishmania infantum population in the resistant strains. And then we got these mutations, we made the pattern, just the profile, and we see that it hits the same 3 nucleotides and the same type of signatures as our signature. So that's one of the ways to validate that. And then in the same year, that was in parallel validated by another group, the group of Edwin Kupen, where they did it in organoids. So if you treat organoids with 5-fluorouracil, you obtain the same signature. So it's very well validated by different ways. And there are many other things in the paper that I'm not going to cover today, but you can read it. I'll just summarize here some of the things. So we identify these mutational signatures of platinum-based drugs and also 5-fluorouracil. We also detected the signature of temozolomide, but only associated to mismatch repair deficiency. We found that these chemotherapy mutations are late. If we can, in the cases that we can measure if they are very early or very late, and they tend to be late very clearly, and they tend to be subclonal. So the cartoon that I show that they are not clonal, but they are kind of in the middle, that's exactly what we found. We also, once we have the signatures, then we can measure what is the contribution of these chemotherapies to create mutations, and we see that it's on the order of 1,000 mutations per metastasis. I'm not showing the results, but you can imagine that this has a huge variation. But the message here is that it's not a few number of mutations. We also saw that the longer the treatment, the higher the number of mutations, and there is a clear correlation here. Okay, that was all studies in metastases, but we are also interested in seeing if healthy cells, not only the tumor cells that are going to do metastases, healthy cells accumulate chemotherapy mutations. And the reason here is that most of these are systemic therapies that may affect any tissue and any cell in our body. But the problem is the same problem that I told you before, right? We cannot just get easily blood or other tissues and just sequence because the mutations are going to be private unless there is a clonal expansion. And then we thought maybe an opportunity is if we analyze secondary leukemias, because these are patients that maybe for a solid tumor were treated, and at that time the hematopoietic tissue was healthy, and later on they developed acute myeloid leukemia related to the treatment. This is T-AMLs. So we have this strong bottleneck at some because of the many cells that die, and also a clonal expansion. And then we thought that there may be an opportunity to see if these healthy hematopoietic cells accumulated mutations of the treatment. So we team up with a hospital in Barcelona, Hospital Sant Pau, and we collected several TAMLs. We sequenced them at whole genome sequencing, and we also collected other whole genome sequence data from TAMLs and AMLs, not related to treatment, from the literature. And we did mutational signature extraction on all these TAMLs and AMLs, and we identified the signature of the cisplatin. If you look at here, every dot here is one AML, and all the AMLs that are related to platinum, so that the patient received platinum drugs, all of them have the signature and on the order of 1,000 mutations, similarly as in the metastases created by with the pattern of the cisplatin. Other TAMLs, but treated with other drugs, we don't see a signal, and primary AMLs not treated, also we don't see the signal. We also saw a higher number of double base substitutions in the platinum-treated TAMLs, which are also known to be caused by platinum drugs. In contrast, we didn't see any trace of the 5-fluorouracil mutations, and there were 3 patients that were treated with 5-fluorouracil. And also these 3 patients, I don't remember if the 3 of them, but some of them at least were treated at the same time with platinum and 5-fluorouracil. So we thought, in principle, no reason not to see the signature, and we were thinking why that could be. And I want to present you here a model that may explain this. So there are very different mutagenic mechanisms of platinum-based drugs and of 5-fluorouracil. So in the case of platinum, it modifies, as the model that I presented at the beginning, it modifies directly bases in the DNA. And then when the cell divides, even if it's before or later after the treatment, like 1 year later, imagine after the treatment, all the damage that is not repaired in the DNA will be converted into mutations. Okay? But in the case of 5-fluorouracil, because what it does is to modify the pool of nucleotides, because it's a timing analog, and this pool of nucleotides, after the treatment, it recovers. And the cell doesn't. So the thing is that if the cell divides during the treatment, the pool of nucleotides is modified and you have the 5-fluorouracil there, and then the cells will accumulate mutations. But if the cell divides later, after the treatment, cells will not accumulate mutations. So it's kind of tempting to think, thinking on this model, that the reason why we don't see the mutations in the TAML is a signal that the cells that do the TAML were not dividing during all the time of the treatment. Okay? So it's kind of a marker of the status of the cells during the treatment. The other thing that we saw is that the platinum mutations are clonal. Here we compare it with metastases from the previous study, and TAML tend to be clonal. So most of the mutations have a clonal, so a variant allele frequency very high. So the metastases will look like this. The cartoon I showed you before, mutations are subclonal. In the TAML, mutations are clonal. If you think about it, what it means is that we are seeing the treatment mutational signatures of one single cell. So a single cell that expanded after the treatment. Okay, so there is a full clonal expansion. What this indicates, the fact that the mutations of the platinum-based drug are clonal, indicates that there is a full clonal expansion from one single cell after the treatment towards the TAML. All these things are represented in this complicated plot, but basically here the message is that chemotherapy mutational signatures can be used as a barcode of clonal expansion and cell state during treatment. Okay? Cell state because If the mutation, in the case of 5-fluorouracil, if the cell doesn't accumulate mutations, it may be one indication that it was not cycling during all the period of treatment. Even if the individual cells accumulate mutations, we will not see them with a bulk sequencing unless there has been a strong bottleneck and a clonal expansion. So the fact that if we see the mutations and if they are clonal is a way of barcoding the cells for this clonal expansion. And the advantage here is that because the treatment, we know exactly when the treatment occurred, and this is a specific period of the time of the life of the patient, this is the reason why it can be used as a barcode. We are not the first one to use the mutational signatures as a barcode, and there is a previous paper by Francesco Maura lab where they use it as well in multiple myeloma in a very clever way. The other thing we wanted to see is if we can detect the mutations in the blood. So now let's imagine all the samples in TCGA that we have tumor and blood, and usually the blood is used for identifying as a control for germline variants, right? To identify the somatic mutations in the tumor. We have the same thing for Hardwick data that are metastases. We have whole genomes and we have tumor and normal blood. So what we thought is maybe we can identify the blood somatic mutations if we do a reverse calling. So we are trying to identify somatic mutations in blood using the tumor as a germline control. And we did that for all Hardwick. We also did it for all TCGA. And in the case of Hardwick, because we have whole genomes, we have quite a lot of power to identify mutational signatures. And we identified a good number of blood somatic mutations. And this is an indication of clonal hematopoiesis, because if we can identify them, it means that they have a high variant allele frequency and they are in clones of clonal hematopoiesis. We extracted mutational signatures, and it was very reassuring to see that we find the hematopoietic stem cell signatures. So many of these mutations are real hematopoietic mutations. But we didn't find the signature of chemotherapies, not even the platinum-based signature. And these are the same patients, Hardwick patients, that we already find these signatures in the metastases, but we do not find it in the blood. And then we thought maybe it's a lack of power, because in the blood we are identifying a lower number of mutations. And what Ferran did is a synthetic analysis to see if it was a matter of power. And he proved that it's not a matter of power. So that even with a few mutations created by the cisplatin that we are able to identify, we should detect a signal of the cisplatin if it was there. And we used the MSIC-ACT by Rosen Lab. That is a way to test if there is the signal of this specific signature. And the conclusion here is that it's not due to a lack of power, but we really don't see the signature of the treatment in the clones that have been expanded. And one more piece of information that is interesting is that chemotherapy is associated to clonal hematopoiesis. What it shows here is that the patients that have gone through a cytotoxic treatment, they have more frequently clonal hematopoiesis, and also of a specific mutations in specific genes like CHEK2 or PPN1D. Right? This is our data, but this was also shown earlier by Ellie's lab, Ellie Papa-Manuel lab, and Kelly Bolton. So what we see here is that chemotherapy selects pre-existing clones with specific mutations, because the fact that we don't see the signature of the cisplatin in patients treated with cisplatin, it means that they are not in the clones as we saw in the TAMLs, right? That all the cells of the TAML have the mutation and it's the mutational signature and it's clonal mutations. In the case of clonal hematopoiesis, we could identify the signature if it was there, but the fact is that the chemotherapy mutations are private to a number of cells and not in the clone. So this is indicating that the expansion of this clonal hematopoiesis, these clones, is not posterior to the treatment, but is anterior to the treatment. So in this cartoon, we show the message here, which is that we have clonal heterogenesis in the bone marrow with some small clones already. At the time of the treatment, there is this bottleneck, and when there is a TAML, all the cells derive from one single mutation after the treatment. Well, when we can identify clonal hematopoiesis, even related to the treatment, because we see these clones associated to chemotherapy treatment, but they are from pre-selected clones that were already there and have been selected due to the treatment. Okay, so summary, I show you about the mutational footprints of chemotherapies, that we can also use these chemotherapy signatures as a barcode of clonal expansion and cell state during the treatment, and the evolution of hematopoietic cells under cancer treatment. And I'll finish here and take questions.
Thank you, Neria. That's a very nice talk. We do have some questions. This from Emery Kokakavuk. Can the absence or presence of the chemotherapy signature tell us about the efficacy of cisplatin or 5-FU therapy for individual patients?
About the efficacy, okay. Oh, that's interesting, maybe. So I think that what it tells us is about how strong is the bottleneck and the clonal expansion, and that actually could be related to the efficacy of so we saw also that is related, for example, if the metastasis is a distal metastasis or if it's a proximal metastasis, we also saw this relationship. So it shows this efficacy. I think it can also indicate some of this, but we didn't test it. It's a good point.
From Dimitri Gordinin, could the physician's decision to apply cancer therapy, which should account for individual patients' features, affect mutation spectra and metastasis regardless of a specific treatment?
I'm sorry, I'm not sure I understood. Can you repeat?
Right. So, yeah, I can repeat it. Could the physician's decision to apply cancer therapy, which should account for individual patients' features, affect mutation spectra and metastasis regardless of a specific treatment? Okay.
So I assume it asks if because that we see differences. So I say very quickly that we see on the order of 1,000 mutations, but there is a lot of variation here. So there is variation between patients. And that's something that we don't understand. There may be many reasons why different patients accumulate different number of mutations, and I can imagine in the future that would be something that you take into account at the time of deciding if you treat a patient with, not yet, because we don't understand and we don't know how to select patients at all, but hopefully at some point we will. And for some patients, that chemotherapy is probably more toxic in terms of mutational toxicity than for other patients. And if at some point we can distinguish those, it could be to take it into account. We are not there yet.
Okay. Yeah, I have a question. So can you use, what's the potential for using mutational signatures to monitor chemo treatment? And perhaps like assaying liquid biopsies from circulating tumor cells that perhaps could be expanded so that you could have enough cells to assay?
Yeah. So to monitor, I mean, one way that you could monitor is, because I always said that if we don't have a clonal expansion, we cannot see it. But actually now we have some techniques that we could see it. And you could monitor in some patients in the blood if there are mutations, like, for example, using nanoseq, duplex sequencing, something that you can identify mutations at very low variant allele frequency and still trust them. And you could measure, for example, what I call mutational toxicity of the chemotherapies. This will only see the mutational toxicity, not if the treatment is being effective or not for the to stop the cancer. But yeah, there would be some ways that you could monitor these kind of things.
Okay. And one more question from an attendee. Did whole genome sequencing of TAML reveal any recurrent patterns of structural variants associated with treatment? That is, genes involved interstitial deletions or chromosomal translocations, etc.
Yeah. Yeah, I didn't present it here because I focused on the mutational signatures, but yes, these TAMLs, they have some of the usual drivers, and some of them are translocations that are very frequent in TAMLs. And we can also see them in these patients. And what we don't know if what's exactly the relation between the treatment and the creation of these translocations. I mean, this is, I think, a bit more clear in the topoisomerase inhibitors, that we always see the same type of translocation. But in cisplatin, to be honest, I don't know how, because cisplatin also creates some instability, and some of these translocations may also be caused by the treatment, but somehow they are selected, and we see very often the same ones or hitting the same genes.
Okay. Well, thank you again for a wonderful talk, and we'll move to the next speaker, and that is Dr. Hannah Carter from the Division of Medical Genetics at the University of California, San Diego. And her talk is Interactions between common immune variants and the tumor genome influence immunotherapy outcomes. Dr. Carter?
Thank you for the introduction, Ron, and I'm very excited to be here. And thank you, of course, to the organizers for the invitation. So our lab is interested in cancer precision medicine and using information from the genome to try to improve patient outcomes. And we kind of think about two areas here: the germline genome or inherited genome, which is typically used to try to project lifetime risk of developing cancer, so we can identify individuals at high risk and target them for aggressive screening and early detection. But once an individual develops cancer, we think a lot about how we can use the somatic genome to try to map them to the drug that's going to give them the maximum benefit. But we think a lot about tumor evolution and selection. And one thing that we've been thinking about a lot lately is how genetic background might influence the evolutionary trajectory of tumors. Of course, this is a challenging problem to study because tumor genomes tend to be quite heterogeneous, with few tumors sharing mutations in the same genes. Nonetheless, we can recognize tumors because they develop these similar characteristics. And that sort of suggests that there are these underlying pressures that, that basically force tumors to evolve in certain ways and optimize certain criteria. For example, resisting cell death, accumulating mutations in genomic instability, and evading the immune system. And I'll focus on this later. The fact that all tumors develop the ability at some point to evade the immune system suggests that the immune system is imposing selective pressure on developing tumor populations. And if we can impair the mechanism by which the tumor cells are evading immunity, then we can get T cells to now effectively eliminate tumor cells. So this was one of the first places we decided to try to dig in for more of these types of germline somatic interactions during tumor development. And the first place to look here is a molecule called the major histocompatibility complex. So this is sort of an obligate molecule for immune surveillance, and its job is typically to present peptides on the cell surface for surveillance by T cells. And in general, the T cell repertoire should be depleted for T cells that can react to self-peptides due to a process called thymic tolerance. But in the context of cancer, if you have mutant proteins and a peptide harboring the mutation makes its way onto the cell surface, that mutation is not expressed in the thymus. T cells will not have been pruned, and they can potentially recognize this as a foreign molecule now and induce or execute a cytotoxic program against the tumor cell. And a number of different immunotherapy strategies rely on this interaction, including neoantigen vaccines, immune checkpoint blockade, and adoptive T cell therapies. So that kind of makes this molecule very important in the context of immune surveillance and immunotherapy responses. If we think about the germline aspect of things, this molecule is actually quite heterogeneous in human populations. I'll just explain quickly. There's actually two classes of MHC. Class 1 is expressed on all nucleated cells and presents predominantly shorter peptides from endogenous proteins on the cell surface to CD8 T cells. I'm going to focus mostly on class 1 in this talk, but class 2, which is expressed predominantly by professional antigen presenting cells and presents mostly peptides from exogenous proteins that have been endocytosed, and presents longer peptides is still really important. You need these activate CD4 T cells, which provide help to the CD8 T cells to generate effective anti-tumor immune responses. If we look at the genes encoding the MHC, these are the human leukocyte antigen genes in the HLA region on the short arm of chromosome 6. Class 1 is the peptide binding group specifically is encoded by 3 genes, HLA A, B, and C. It's a bit more complex for class 2, but for both of these, there's this huge diversity of alleles in human populations, and that's because these molecules are in an evolutionary arms race with pathogens. Nonetheless, these different alleles can have different exonic sequences, and that can result in sort of differences in the composition of the peptide binding groove of the molecule, which can confer different specificities for peptides to different MHC molecules. So thinking about this in the context of cancer, you can imagine that any individual is going to have 2 copies of HLA A, B, and C for MHC class 1. And this is going to dictate the space of peptides that can be subjected to immune surveillance. Now we can intersect this with cancer-causing mutations, and you can imagine that there could be cancer-causing mutations that can be incorporated into a peptide that would be effectively exposed for immune surveillance by this particular genotype, but there could also be cancer-causing mutations that would never generate in a well-presented peptide and thus would be effectively invisible to immune surveillance. And if this is happening, then we can consider that these exposed peptides would more often result in immune elimination of the harboring tumor cell, and thus would be observed with lower probability in tumors, whereas these invisible mutations would not be pruned by the immune system and should persist. And so to test this hypothesis, we developed sort of a computational framework that seeks to evaluate the potential of each allele that an individual carries to generate a peptide that would be effectively presented overlapping a particular mutation. And so individually, the highest affinity peptide is selected for each individual allele, but then all of the alleles together have the potential to present one or more of these peptides in the cell surface. So we aggregate the scores across into a single score, which we call the patient harmonic best rank or PHBR score. And in this context, a lower score is more is associated with more effective presentation by the MHC, and this is validated against mass spec data sets to show that indeed you recover more true presented peptides in mass spec data when you focus on these lower scores and lower ranks. Then we applied this to the TCGA across 10,000 tumors, focusing on driver mutations that we think would have occurred earlier during tumor development, because after the tumors evade the immune system, then this relationship should break down and it should no longer matter if a peptide is being exposed or not. And amongst these more recurrent driver mutations, we found that indeed having a higher PHBR score or poorer overall presentation was associated with increased probability of observing these mutations in the tumor. And this was not the case for sort of one-off passenger mutations, which are probably predominantly occurring late in tumor genesis, and then nor for germline variants, which should not be subject. They're subject to thymic tolerance, and therefore you shouldn't get T cell reactivity against them. Okay, so this suggests that this paradigm might be holding up. We can then ask, does it hold for multiple tumor types? And you can see that the relationship between the probability of observing a mutation and the PHBR score seems to generally hold across tumor types with some variation. Interestingly, we see kidney cancer down here doesn't seem to show an effect. Some of these may be due to small sample size, but there is for some tumor types evidence that the tumor immune interactions are quite different in certain settings. And then for some of the larger effect sizes, we think maybe they're inflated because there's a particularly recurrent driver mutation that is generally poorly presented in those tumor types, such as thyroid cancer and low-grade glioma, where you have a lot of BRAF V600E and IDH132 mutations. So if we think about this as it relates to mutation frequency, we can see something I think interesting for considering, highly recurrent driver mutations as immunotherapy targets. And so specifically here, we've binned these driver mutations according to how recurrent they are in human, in the TCGA data set. And so BRAF V600E is going to be down here in this, over 600 tumor mutation category, but some of these are only seen mutated in 2 or 3 tumors across the data set. And here this heat map is has a patient on every row and the potential of their MHC genotype to present the mutations in this particular bucket. with the darker color indicating more effective presentation. And so what we can kind of see is that on the left side, where the mutations tend to be less seen less often, we tend to have slightly darker colors, indicating that on average, more MHC genotypes are able to present these reasonably well. And then on this right side, we see that the color is getting lighter, sort of suggesting that there are some of these more recurrent mutations are just generally less effectively presented by MHC across all of the patients. And so what that might suggest is that part of the reason some of these mutations are seen so frequently across tumors, it's not just because of the fitness advantage that they provide, but also potentially because they are more difficult for the immune system to pick up, which has, I guess, a couple of implications. One, it might be more difficult to target some of these highly recurrent driver mutations with immunotherapy. And the other is that, maybe there exist driver mutations that are infrequent because they get picked up more often by immune surveillance, but nonetheless may have very strong fitness effects in the subset of patients that carry those driver mutations. So if we want to continue thinking about potential implications for immunotherapy, we can look at this in more depth in the context of immune checkpoint inhibitors. And so the idea here is that when a T cell detects the MHC presenting in a neoantigen, it wants to attack the tumor cell, but it has these co-inhibitory molecules that are expressed on the T cell. And tumor cells often begin to express ligands for these co-inhibitory receptors, which causes T cell exhaustion. This can also be expressed by some of the infiltrating immune cells like macrophages in the environment as well. But if you can introduce so these antibodies that block this interaction, then you can reinvigorate the T cell and get it to kill the tumor cells again. And early studies showed that the patients that got the most durable clinical benefit from ICV treatment tended to be those that had a higher burden of non-synonymous mutations in their tumor. This is from the Rivzi Science paper in 2015, but this has played out over multiple tumor types, with some of the greatest benefit being observed in melanoma, where we have a high burden of UV-induced mutations, and in mismatch repair deficient tumors, where there tend to be a high proportion of sort of indels and frame shift events that might create particularly potent antigens. Nonetheless, even those tumors tend to have sort of a lower objective response rate than we would like. And so there's a lot of work going on to try to understand this and to improve outcomes. So although initially we see that the tumor mutation burden seems to predict outcome, thinking about it in the context of an individual's MHC genotype and antigen presentation, probably the high mutation burden is just a proxy for having more effective neoantigens. And so we tried to investigate this in a cohort here at Morse Cancer Center with Rizal Kurzrock and Aaron Goodman, work done by Andrea Castro. And we looked at 77 tumors that were treated with checkpoint inhibitor monotherapy from a variety of tumor types. And they had somatic mutation profiling here with Foundation Medicine, which would give an estimate of the tumor mutation burden and a subset of the mutations. However, we note that this should the subset should include the majority of driver mutations, because it's going to include many of the key cancer genes. We were able to replicate that a high tumor mutation burden was associated with better progression-free survival in this cohort as well, and then asked, how does MHC genotype play into this? And what we found was that amongst the patients that had a high tumor mutation burden, those that also had a really well-presented mutation on the panel based on their MHC genotype had the progression-free survival benefit, whereas high tumor mutation burden in the absence of one of these really effective neoantigens was more like these low tumor mutation burden tumors and generally had worse outcomes. So this is sort of in line with the idea that it may be the quality, not the quantity of the neoantigens that matters for immunotherapy responses. And there's some related work by Diego Chowell looking at HLA diversity. So individuals that have a homozygous HLA type or carry multiple alleles that have very similar peptide specificities tended to gain less benefit from immunotherapy than individuals that had sort of a broader array of peptides that could be presented by their HLA genotypes. So of course, there's a lot of interest then, how do we map mutations to immunogenic neoantigens and do a better job projecting who's going to benefit from immunotherapy? And there have been a number of factors that have been identified related to immunogenicity, though it's still an open problem and there's a lot of false positive predictions. So we're still trying to figure out why we think something should be immunogenic, but they never are. But some of these relate to the accessibility of the peptide and others relate to the quality. And so in terms of accessibility, we have things like the level of the mutant allele expression. The more abundant the mutant protein is, the more likely the peptide is going to make its way to the MHC. It still depends on proteasomal cleavage of the peptide, top transporter integrity to get it into the endoplasmic reticulum. And then we've recently found that subcellular localization can predict to some extent immunogenicity of peptides. If you're interested, you can check the paper out on bioRxiv. Quality-wise, we think about affinity for the MHC, agrotypicity, which is if the wild type is well presented in the thymus, does that reduce the chance that the T cell repertoire is going to be sensitive to the mutant peptide? The stability of the MHC peptide complex, which can relate inherently to the MHC allele itself, and foreignness. So the more different the peptide looks, the more likely it's going to generate TCR reactivity. But as I mentioned, we're still kind of looking for what factors are going to predict effective immunotherapy response. And so here, there's an interesting work by Cummings et al. That starts to look at mutational signatures as relates to this. And so what they noticed is in a paper by Chow et al., it was found that melanoma individuals with the B44 supertype, which is a set of like 4 HLA B alleles, had better outcome in the context of immunotherapy, but they could not replicate this in non-small cell lung cancer patients. Those patients with the B44 supertype actually had worse outcomes when treated with ICB. In the Chow paper, it was noted that the responders to ICP tended to have a higher proportion of these radical glycine to glutamic acid missense mutations. And they were actually able to see that when they compared the proportion of mutations leading to these types of glutamic acid substitutions in their cohort to those in melanoma, that the melanoma patients tended to have a much higher proportion of these. And they were able to tie this back to the mutational signatures associated with melanoma and smoking, respectively, with this high proportion of C to T transitions happening with UV radiation, generating these negatively charged substitutions that would then have high affinity for B44, which has a positively charged binding groove. And they identified one other B supertype that has a charged binding groove, which is the B27, although it's the opposite charge. In any case, they speculated that these mutations that were creating these effective neoantigens for B44 were driving the response in melanoma. So they partitioned their non-small cell lung cancer cohort into the individuals that carried one of these substitutions to glutamic acid versus those that had the B44 supertype but did not. And what they found was that indeed you could now stratify the patients such that those that had the B44 supertype and had the motif had a better outcome. And here they're showing the same for the combined B27 and B44. So this suggests that the mutational signature may also interact with the MHC genotype to help predict which patients could potentially have effective neoantigens and benefit from immunotherapy. Now, perhaps a cautionary tale here, we also see frequent somatic loss of the MHC as a mechanism of immune evasion with more frequent mutations in HLA-A and B, but also loss of B2M, which is a scaffold that is required for MHC formation. These tend not to affect the peptide binding groove, but instead the interaction with the T cell receptor, the transmembrane domain, or the signal peptide, and they seem to predominantly affect A and B, not so much C. But when we look at patients that have these somatic mutations, they tend to have a higher burden of non-synonymous mutations, a higher fraction of mutations that should generate neoantigens. And I should note here, interestingly, this is just in microsatellite stable tumors. We had to remove microsatellite unstable tumors because they have this huge enrichment for B2M and HLA mutations. And so that may be an even more common mechanism of immune evasion in these tumors that that have the MSI, and maybe that's also a place where we should be looking at potentially mutational signatures interacting with mechanisms of immune evasion. If you look at the allelic fraction percentile within tumors, sort of like how much of the DNA harbors these events, B2M mutations look like they're pretty prevalent and maybe are early or dominant mutations in the tumor, whereas HLA had sort of this bimodal distribution. And when we compare those that have sort of a dominant HLA mutation that is present in most of the DNA versus those that look like they might be more subclonal, we could also see a higher burden of these putative neoantigens and sort of these cases where there was a higher prevalence of HLA loss. And so that sort of suggests that basically the mutations to these are disconnecting immune surveillance and allowing tumors to accumulate these mutations that should otherwise have resulted in elimination by the immune system. This is consistent with work by McGranahan et al. That show loss of heterozygosity frequently in some tumor types, estimating up to 40% of lung tumors have this, but it may be more variable across tumor types. And notably, loss of HLA and B2M has been shown to contribute to poor immunotherapy response and resistance to immunotherapy. So even if you do have alignment between mutational signature, et cetera, it will only do you some good if you have the MHC intact. So since I'm running a little bit late, I'll skip this last section. It has less to do with mutational signatures and more to just say more broadly, the germline influences immune traits, and this is related to cancer affecting both aspects of innate immune responses and adaptive immune responses. And in particular, I think TREX1 is associated with single-stranded DNA. And so maybe this is another place we should be looking for potential interaction with mutational signatures and immune responses. So just to conclude, the inherited genome is influential in host anti-tumor immunity with a major dependency on the MHC, but also other aspects of immunity. And the mutational signatures are going to interact with this and potentially will be informative for response to immunotherapy in the future. So thanks to my group and my funding, and thank you for your attention, and I'll stop here.
Okay, thank you, Hannah. That was a very interesting presentation. And attendees can ask questions through the question and answer or chat box. I will start. So you talked a lot about variation in presenting the epitopes through the MHC. I'm just wondering, on the receptor side, is there variation in terms of between individuals of let's say T cell receptor interactions that might also affect, in recognizing the MHC epitope that would affect these responses?
Yeah, absolutely. But I think it's an astronomically hard problem because the T cell repertoire is huge, and at any given time, it's not clear that all possible, recombinations are being expressed. And then on top of that, different TCR sequences may recognize an array of different antigens. It's very hard to figure out which specific TCR recognized a particular antigen at any time. So it's hard to track down exactly what the interaction was.
Yeah, I guess in addition, I was kind of wondering if there were kind of generic processes that affect TCR maturation that might also play a role.
Yeah, no, that's a good point. And I think there probably are. I'm less familiar with the TCR side of the equation, but I think that's also worth looking at.
Okay, and just one other question that's come to mind. In terms of neoantigen presentation, in terms of the DNA that's, where the DNA where these epitopes arise, is the genomic distribution widespread? Are there any biases? Like, are there particular spots or biases in the areas in the genome where neoepitopes arise, or is that just pretty much randomly distributed throughout the whole exome?
So there was a paper from Fritz Roth's group that showed that you can pick up the sort of the selection looking just at the expressed regions. So I think, the expression of the mutations is really important in terms of them being able to serve as a new epitope. But we're also seeing now, I think, that the subcellular location of particular proteins might be influential. I don't know about like the specific patterns in sort of the 2D genomic space, if there's like a particular 2D regions that are enriched, other than outside of it being in particularly regions with a lot of peptide or proteins that would generate peptides effectively.
Or perhaps in cell cycle, like early replicating genes versus late.
Yeah, that's an interesting idea. We could look at that. I know there's some sort of a higher error rate in terms of mutagenesis and genes that are late replicating.
Okay. All right. Well, thank you again. And we'll move to the last speaker of the session. And that is Adam Schlein, who's in the Department of Laboratory Medicine and Pathobiology at the University of Toronto. And the title of his talk is Clinically Relevant Mutational Signatures in Childhood Cancer. Adam?
Hi, everyone. Let me just try and get the timer up and hide my own face. Okay, nice to be here, and thank you very much for the invitation. This has been a really great couple of days, really, really interesting. The way I sort of interpreted my job here is to prove to you that there's a high and broad relevance of mutational signatures in the context of childhood cancer, and that we can learn pretty broadly from childhood tumors across the age range. So cancer, unfortunately, is still the most common cause of disease-associated death in the early years of life in the developed world. In Canada, with a population of about the size of the state of California, there are about 4,300 new kids and young adults who are diagnosed with cancer per year. Of these, about 30 to 40% will relapse, in which case the survival is really low. I mean, in these Kaplan-Meier curves shown by, developed by Stefan Pfister, you can see whether it's brain tumors, bone cancers, or liquid cancers, the survival is really abysmal at relapse and upon metastasis. And this is on top of the late effects and morbidities caused by the often toxic chemotherapies that these kids are given. And for this reason, and because of the relative rarity of childhood cancer, we have to work together, create consortiums. We created the KICS Cancer Sequencing Program at SickKids, and I'll describe some national efforts in Canada as well to sequence and look deeply at these patients' tumor genomes and their germline genomes. Childhood cancers also provide a relatively unobstructed view of mutational processes across all age ranges. We know that they're known to be relatively quiet, although I'll show some examples where that's not always the case. They therefore have few background mutations, and there is a limited repertoire of extrinsic mutagens that these children have hopefully been exposed to. And so the overview of the talk is really going to be two main stories where I've taken, for the past few years, really extreme examples in childhood cancer. One in the context of inherited mismatch repair deficiency, or CMRD, as a model of early onset tumorigenesis. And then looking at using that as a way of, or a window into understanding the causes and consequences of hypermutation across the age ranges, and looking a little bit at immune checkpoint inhibitor therapy and developing new biomarkers. And then I'll switch gears to talk about diagnostic gene fusion formation, which is often a cause of many childhood cancers, and use that as an entry point to talk about genomically complex muscle cancers. And we'll finish off by telling you about some practical uses and hopefully lead to some discussion about practicalities of mutational signatures for ongoing patients. Put this on here. Right, so first Gadi actually told you a little bit about this very penetrant, devastating syndrome called constitutional mismatch repair deficiency, involving two hits, usually one coming from mom and one coming from dad, in MSH2, MSH6, MLH1, or PMS2, most frequently PMS2. And it leads to a slightly different spectrum of tumors that you see in Lynch syndrome. It does have the colorectal cancers, but also brain and leukemias and other cancers, with a median age of onset of only 7.5 years. As I said, very penetrant. With Uri Tabori, we studied and determined that these tumors are ultra hypermutated, often with mutation burdens of approaching 100 to 500 mutations per megabase, compared to most childhood brain cancers that are quite easily differentiated, as well as childhood cancers generally. There was a mutational signature that we saw, really, I should say, a pattern back then in 2015, which we thought related to the acquisition of the somatic POLE and POLD1 mutation with the inherited underlying MMR. But what we saw, well, specific wasn't really quite satisfying. And so we dug deeper by looking at the then 81,000 patients in the Foundation Medicine cohort, sequenced through a panel. to help them and to look at the burdens of mutations to determine thresholds for hypermutation, and to look at the causes and consequences of those hypermutant tumors, both in kids and adults. We found at the time that there was a surprisingly broad array of childhood cancers that were hypermutant, above 10 mutations per megabase, including sarcomas, nephroblastomas, and many others, up to about 5% of childhood tumors. And within this band of 10 to 100 mutations per megabase, we saw tumors that had some amount of mismatch repair deficiency, with or without POLE, POLD1, whereas here, these tumors are almost exclusively those that have a combination of those two different hits. We later learned that this, you should never say exclusive, that these things are not exactly, we've learned more about these ones that are ultra mutated, and I'll show you some more recent data of different causes of ultra mutation in childhood cancer. And when we clustered the Foundation Medicine cohort based on their mutation burden, based on their trinucleotide context, you see 8 major causes of hypermutation at the time across this cohort of heavily mutated tumors. I'll just point out there's some major things that one sees here, but a much broader activity of major mutagens. For example, amongst the UV light group, what we call it cluster 6 over here, there are of course many melanomas, but there also were angiosarcomas, which are a group of tumors that has since been recognized as being hypermutated and even responsive to ICI. We were able to help to re-diagnose some of the cancers on the basis of the mutational signatures. The clinical annotation, for example, of some of the lung cancers was that they were lung cancers, but in fact they were probably misdiagnosed metastatic skin cancers on the basis of the mutational signature and other features that we examined. But looking at the MMR clusters, 1, 2, 3, we see differences in mutation burden and differences in signatures on the basis of the age of the patients and the POLE presence or absence of a POLE exonuclease domain mutation, as well as massive differences in the spectrum of tumor types. The cluster number 2 are really those sporadic colorectal cancers and others that have combined, that are usually considered to be classically MSI high, and clusters 1 and 3 are those that have a POLE mutation. Whereas cluster number 1 specifically are those patients that have the CMMRD. That's telling you that looking at the somatic genome alone as a readout can help you to what Uri likes to call a reverse diagnosis, a reverse genetic of an underlying condition, which is obviously has implications for that child, their siblings, and potentially the rest of their family. We've used this system successfully to reverse diagnose underlying genetic conditions. Here from this paper showing about 15 patients for which the germline was assessed formally using CAP-CLIA germline panel, and 100% were confirmed to have those PMS2 and other mutations. Many of these patients have then gone on to clinical surveillance to find tumors earlier with great benefit and or immune checkpoint inhibitor therapy with some success. And we were interested, this is from our earlier paper showing that there's a bit of a plateau in terms of the mutation acquisition, where it seems like tumors can become laden with mutations, but up to a certain point. And we've asked ourselves recently whether that genomic plateau corresponds to a transcriptional one, where you can see, does it mean that if a tumor is genomically hypermutant, are they necessarily transcriptionally hypermutant, and vice versa? So specifically, we wanted to create a method to look at somatic transcriptional output. And as I mentioned already, are there cancers that are genomically hypermutant but not transcriptionally hypermutant? Okay, so I'll switch gears a little bit to move away from hypermutation genomically and transcriptionally to talk about formation mechanisms of translocations. And starting with Ewing sarcoma, which is a childhood cancer, quite rare, involving EWS and FLY1. So a gene on 22 and a gene on 11. Sometimes it'll be EWS and ERG. Big question that Nathan, who was a PhD in my lab, student asked was, how and when do these fusions arise? What Nathan uncovered looking at the genomes was that about 40% of the tumors had these chromoplectic loops, driving and bringing EWS and ERG, or EWS and FLI, together, but also impacting multiple other friends across the genome until kind of bringing it back to the point of departure. This is something that's been described in prostate cancer and is found and almost exclusively at the junctions of the driving fusions in Ewing sarcoma. Moreover, when looking at the paired relapse or MET samples, of which we didn't have very many at the time, we saw disappearing mutations suggestive of parallel evolution, which are a mutation present in the diagnostic specimen and absent in the relapse and vice versa, even upon deep sequencing. And using signature 1, and we see a clock-like pattern as would be expected, and the rate of mutation acquisition is about 10 per gigabase pair per year in Ewing sarcoma. And you can see the parallel evolution of this relapsed tumor compared to the diagnostic specimen and the number of additional mutations that were acquired. And using the clock, we can infer that these two tumors diverged from each other from a most recent common ancestor, about 1 to 3 years pre-diagnosis. And in the life of a teenager, a young teenager who has Ewing sarcoma, 1 to 3 years is quite significant. And so what that means is when the child comes into the hospital for their diagnostic tumor, there is something else that is clinically undetected that had begun to grow about 3 years before the patient was diagnosed, and that is the often fatal disease. Looking more recently at leiomyosarcomas, this is a more common sarcoma, tumors that are essentially grouped together on the basis of them all being smooth muscle cancers. We know already that there are massive differences in survival on the basis of site, and the survival at relapse is very low. This time, looking at both our data and TCGA, and supplementing this with multiple different time points and multiple different sites within each individual tumors. Unlike Ewing sarcomas, the leiomyosarcomas are quite large, allowing us to do this multi-site sequencing. We could ask again when and also how did these sarcomas originate? And with Rebecca Glady, a surgical scientist at Sinai in Toronto, we could also create these trees, again, based on the multiple different sites, multiple different time points, to infer the time of divergence from the most recent common ancestor. And so I won't go into massive detail except to say that the divergence from the clone that will become the relapse in these individuals was 27 years before diagnosis. And then for the secondary MET in this individual to their thigh was 20 years before diagnosis. And because we had multiple different sites, sometimes 1 to 2, 3 centimeters away from each other, we could time the divergence of these from their most recent common ancestors to be somewhere between 1 and 6 years before diagnosis. There have been some suggestion early on, mostly based on exome sequencing, that there was a very high proportion of LMS tumors that had signature 3 or BRCA-NES. We evaluated this, bearing in mind a lot of the challenges that were described yesterday, and see that indeed there is, maybe not to the degree that was described in the past, some evidence of HR deficiency to the tune of about 64% having either the sub phenotype or the concordant indel signature. And being cautious about this, we also did in vitro validation with talazoparib, a PARP inhibitor, to show sensitivity in a panel of LMS lines. Okay, I don't really know how much time I have left, but I'll keep on going and try and get to some of the more practical aspects of things that we're doing right now. So we are, I've undertaken a precision medicine program, as many institutes have, here using deep panel sequencing combined with whole genome, the blood in the tumor, as well as RNA-seq, to enroll children and young adults that have hard-to-treat cancers. Importantly, we also include patients with cancer susceptibility, for which nothing has been found so far by conventional methods. We've also linked up with 2 other sites across the country that are also doing sequencing and have opened up a program called Profile that will enroll children and young adults with these hard to treat or hard to cure cancers. Really, no matter where a child is in the country, they are eligible for this program with over 20 participating sites involved. Importantly, we're doing genomes in addition to the deep sequencing and the RNA-seq. Okay, so I may have gone pretty quickly, but I'll conclude with 5 major points, kind of summing up a lot of the things that I've showed you already. And hopefully we can use this as a point of discussion later on, the high clinical utility of mutational signatures. One, in terms of cancer risk, uncovering these relevant germline mutations, using the tumor to help to diagnose the germline variants. I haven't shown you the data that we have on variants of unknown significance, but the tumor signatures can also help to resolve those. We can differentiate metastatic cancers from new primaries to help us in diagnosis. Also looking at chemo-induced secondary cancers, which are a big problem in childhood tumors. We can sometimes suggest HR deficiencies, which would give us a new therapeutic option, as I showed in leiomyosarcoma. We see evidence of drug resistance, including these markers of prior therapy. And we can just use this clock-like signatures to time the divergence of metastatic clones and potentially in the future to be able to intervene prior to relapse. With that, I would just like to thank everyone in the group who's done this great work, and to our funders. Thank you for your attention.
Thank you, Adam. We have a couple questions. We have one from Ruben Harris. What are the signatures of the transcriptional level changes?
So I think Ruben is talking about the transcriptional outputs.
Yeah,
I assume so. Yeah. So there are no particular signatures that are involved in the high RNA output that we observed. It's essentially, we don't see a preference for one signature or another. It seems to be just the tumor RNA deciding to churn out a lot more of its transcripts than it would otherwise. And we don't see it to date a signature that.
More transcripts, not different signatures in the transcripts?
No, just more transcripts. It's just sort of doing more with what it has.
Okay, because I was kind of, I guess my mind wandered the wrong direction. I was thinking more in nucleotide imbalances and things like that.
That's a very interesting question. So far, we don't see that, Ruben.
Thanks.
Okay, from the attendee, how does sensitivity to PARP inhibition compare to sensitivity to cell lines with BRCA1/2 pathogenic variants?
It compares quite well. I don't have the numbers at my fingertips, but those BRCA mutated cell lines are positive controls, and the leiomyosarcoma tumors performed just as well as they did.
Okay. Question from Mila Prokunina Olsen. How do you determine the causality of those germline variations beyond the fact that they are identified in patients?
I mean, it's they're identified in patients. We use first of all the rubric that's generally used by CMG and others to determine causality and association and severity, pathogenicity. So they're present in the patients, their absence from the controls, they're at the hotspot domains, and the phenotypic output is one of a massive and very specific mutational signature in the cancer genome.
Okay. I have a question. So you've shown that for tumors that arise from germline variants, carriers with, let's say, mismatch repair, have high mutational burden and signatures. Do you know in germline carriers, are there mutational signatures in the normal tissues of heterozygotes? Thinking that maybe there's a way to identify germline carriers for these mutations just by sampling looking for mutational signatures in biopsiable tissue.
Absolutely. So actually, Gadi showed some beautiful data yesterday using MSI signatures that he could find, maybe not the Lynch syndrome patients, but certainly those with CMMRD, based on the germline test, and based on that sensitivity is quite beautiful.
Okay, great. And I guess just related to that, do you see mosaics with some of these mismatch germline carriers, so that you have that it occurred a little bit later in development?
Yeah, so I guess you're referring to things like NF1 will sometimes present as a mosaic and other, but in the case of these, no, they seem to be typically non-mosaic. In in NF1, one does see that. In Leigh-Fermani syndrome, also you see some mosaicism, but yeah, thus far not really in PMS2 and friends.
Okay, thank you again. It was a very good talk.
Thanks a lot.
And now we will move to the panel discussion. And all the speakers from the session can turn on their videos. This will be moderated by Dr. Ruben Harris from the University of Minnesota. And this is to look at sort of the broader themes and questions around this session topic. Dr. Harris?
Excellent talks, everyone. I think I was all along trying to pull a common theme out of these talks. And one of the common themes might have been host genetics and how that influences not only, the actual patterns of mutation, but the potential impact, the clinical impact, and how they might be treated. So, and that seemed to be a little bit in every talk, right? So how do you, maybe we could divide it into diagnostic and therapy, if you want, for discussion. So how can we learn, is it feasible, for instance, to sequence generally without a disease presenting, and then use that sequence to inform therapy before something happens, for instance, like a tumor develops, like Adam was implying, it may be able to pick it up earlier and avoid it later.
I might start first. It's a good question, and of course it's close to my heart because I like germline genetics more than I like somatic genetics. And I think there are some approaches which are gearing towards this direction, which is called genome first, which is difficult for common conditions like cancer, which might be more easy to deal with, extremely rare and more penetrant conditions when you can identify something and you have some predictive value which is determined by penetrance, because like cancer or cardiovascular disease or some common conditions which are difficult to differentiate between people, you need to have so many factors. You need to wait also a long time before something will manifest. You're not going to test people. Do you have cancer yet? Do you have cancer yet? So it's probably not feasible. But if we have something extremely penetrant and predictive, then it could be tested in at least some populations which already have high exposure and higher prior probability that they might be developing something. So which would be probably dependent on clinical conditions for each cancer to determine what it would be, when is it?
Yeah, and I'd love to hear Adam's opinion on that because, with children, I think there's more of an opportunity to make a lifelong impact, right?
For sure, yeah, there's definitely a lot of motivation to do that in kids. I mean, but of course the question still is who to do it is. Are you going to do it across all children? And there definitely is some work that's happening looking at those that are massively predisposed with highly penetrant syndrome. For example, Leigh-Fermani syndrome, there's really a lot of evidence that shows that clinical surveillance works and detects tumors early. So you can imagine pairing that clinical surveillance with some kind of an assay that picks up the mutational signature alongside the regular clinical tests that are happening. That would be the place to start, rather than probably doing it broadly, or in tumors that where you suspect there's a very good chance that they're going to relapse. It's not necessarily predicting of initial tumor diagnosis, but trying to predict the relapse before it happens.
Mm-hmm.
Predicting it, I find it very, very difficult, but I think it's a bit the case of clonal hematopoiesis that it's not predictive, but it's stratifying patients with a bit higher or lower risk, right? It's not something that you could act on more than probably higher surveillance or but yeah, that would be sequencing the blood, seeing that these clones are there, and this indicates a higher probability for leukemia, for example.
Mm-hmm. So your work, Nuria, was also quite interesting in that the therapy was clearly contributing also to detrimental outcomes. So I'm trying, and maybe all of you could comment, to come up with examples of instances where, for instance, the germline might depict it or inform one therapy and not another. So you're adding fuel to the fire, or to use the double-edged sword analogy, you're using the wrong edge of the sword, right?
Yeah. No, probably. So we see all this variation in the mutational toxicity, and we couldn't do the analysis yet, but I wonder if it's related to some germline variation, for example, on the capacity to DNA repair or checkpoints. Mm-hmm. It could be, and yeah, at some point you could imagine that if we understand this a bit better, some of this information may inform or may help to make the decision of which is the more mutagenic chemotherapy, or maybe not, because it may be very detrimental in this specific patient. We'll see. We still need to learn much more about this variability and also the effect of this mutational toxicity, or this number of mutations. Right? We are seeing for now that a big number of mutations are accumulated in the cells, but what is the real effect of them in disease? We don't know yet. I mean, it's tempting to think that it's related to these late effects, but the reality is that we don't know.
Yeah, and I think Dr. Carter, you had, it's not yet standard of care to be looking at the patient's MHC and HLA haplotypes, in informing this also.
Yeah, no, I completely agree. I think there's going to be a lot to find in this area, both with immunotherapy and other treatments. Certainly, you can imagine that the finding that somatic mutations are interfering with antigen presentation might decrease your enthusiasm for applying immune checkpoint blockade to a patient, for example. I wanted to also highlight some work from a colleague, Gareth Bond, I think, published in early 2021, where a enhancer SNP that increases TP53 binding in the KIT LG was found to modify response to DNA damaging agents in patients with TP53 wild type tumors. And so I do think these things are out there, and it's just going to be a matter of tracking them down. But ideally, we'll have a much better idea of how to use the patient's germline to decide which therapies are going to benefit from the most.
Excellent. I want to remind our fellow panelists here too to chime in and turn on your your volume and your video, or type it in the chat, the Q&A, because I think there is lots of great discussion. So a couple of talks also touched on RNA level a little bit. So Dr. Schlen's talk and Mila's talk, where you can imagine RNA level variation also contributing. And that's why I asked that one question. But I'm wondering whether that's controllable also, or not. It just may, because we're not picking it up. If it is stochastic, we're kind of missing it in the sequence analysis, aren't we?
Ruben, may I add, since you asked panelists, just a quick informational addition on your previous question about recommendations depending on sequencing, recommendations of treatments. It is known, it was established first by Tim Fenton, that specific hotspots in PIK3CA, in 542, 545 residues are enriched in APOBEC hypermutated tumors, especially in he actually looked in all. Now, it is also observed in cell culture that these particular mutations in PIK3CA make cell culture resistant to EGFR inhibitors, which are used for lung cancer therapy. So the simple thought, which may be right or not right clinically, is sequence initial tumor lung, especially lung tumor. And if it is already hypermutated by APOBEC, do not use EGFR inhibitors because you have more chance to get resistance. Sorry for chiming in, but
no, that's a great example, Dimitri. I appreciate it. There is a increased resistance to EGFR therapy with that particular allele, but it's actually few and far between. For the hundreds and thousands and millions of mutations caused by, the process I know best, APOBEC, there aren't that many that are therapy or tumor driving. The one that Dimitri referred to is a great example in PIK3CA. But they're few and far between, actually. And Mila, you mentioned another one that might be, but biochemically, the enzymes hate that site. So it could be selection working on top of, rarer events like that.
Yeah, so that's why I'm advocating look only not only at mutation signatures, but individual mutations as well. It's like a population and individual. There's a huge difference because there are cases, of course, when other factors combine, creating this unique opportunities for this perfect crime, okay, biochemically or not. And so just adding a little bit more about the expression levels. So expression levels are transient, and transiency is our enemy because we measure something we can measure, and we are making big conclusions about everything else, but we don't know what was the level of expression at the time when it happened. Like we are not in the room when it happened, and we have to deal with that. So I hate to assume that we know too much about levels of expression in their causalities because we just don't know.
Yeah, no, I agree. And at the end of the day, a signature is the product of the damage and the escaping repair. And we don't know when they all happen. You can generally group early, late, but we really don't.
Mila, could you just clarify that FGFR mutation that you pointed? Is it still a mutation in cytosine or it's in another? It's in cytosine. Then it could be APOBEC. So APOBEC is, imagine an alcoholic that is also a wine drinker, gourmet wine drinker. Alcoholic will drink any cytosine, but will prefer vintage signature cytosines.
Yes, good point. So
and we see it in yeast that are right behind me.
I was wondering about this high RNA output from Adam, and now if it's transient or not, right? The level of expression of particular genes probably it's very transient and very dependent on the time. I don't know if this high RNA output is something more constant, or that probably you don't have the answer, and if you have any idea how it's caused. What's the mechanism?
Yeah, so the best way of looking at the transiency or not is through single cell. We have some single cell multi-site sequencing, and we see actually that there are some clones that seem kind of fated to always pump out lots of transcripts so that they always have high transcriptional output. And those are the clones that express high metabolic activity and have certain other drivers of high transcriptional output that we've been able to pull out. In a sort of a not interesting coincidence, it happens that the Ewing sarcoma as well, that the ETS families, which my rest of my lab is also interested in, are also responsible for de-repressing a lot of the transcriptional output. So when you see higher levels of ETS family, you often see higher amounts of RNA across the transcriptome. So it's quite predictive.
Okay. Yeah. And this is across many different transcripts and even normally not expressed transcript, like
no. Usually what it does is it takes the transcriptional program that already exists and just turns it up. The method is limited by, we have to have somatic mutations in those transcripts to be able to assign them as coming from the tumor versus the normal. And so we can't say definitively that it's every single bit of the transcriptional program, but it looks like it's not taking any special genes and turning them on, but rather just turning everything up.
And I would add also a little bit the question is of high is also very relative. So what is enough? You know, because if you need 5, for example, and getting 30, maybe it's already overkill. It's really not biologically more meaningful than changing from 1 to 5 compared to 1 to 30.
Yeah, I wanted to, just generally comment on the mutability of childhood cancers versus adult. And I think, you dispelled the myth during your talk, Adam, about some childhood tumors being mutationally cold. And there's quite a few examples of mutationally hot ones. And so the word APOBEC was in a couple of your slides too, or at least one. And so I'm wondering where that fits in the spectrum, because at least I was under the impression that it predominantly affects adults, and that raises the question of more general question that you could all comment on, of why some processes are confined to adult and others to children, and vice versa, and not, general. Most should be general, right?
I mean, I could start and just say that we're with Ludmil looking now at the APOBEC signatures across childhood cancer. We've already looked in those tumors that have chromothripsis, and then as kind of predictably, you see the APOBEC mutations closer to the spots of those contagious events. So they don't, I can't really comment on the burden of it right now, whether it is actually depleted overall in childhood tumors. Maybe others have the data to say so.
Okay, but what about the generality of mutational processes specific to one or the other, children or adults?
The processes that need time, like signature 1, signature 5, that's kind of obvious. But other processes like APOBEC, for example, is not that obvious, right? You also need time, but it's very sudden, and it's what we see for the chemotherapy mutational signatures is that the level of mutations, because we have also analyzed childhood leukemias treated with chemotherapy, and the level of mutations is more or less the same. We don't see a difference there. So due to the treatment, they accumulate a similar number.
So the genomes can tolerate or can accumulate similarly high levels, child or adult, that associates with the dose of therapy given.
Yeah, we don't have a lot of data for kids, but the data that we have, it lies more or less in the same mutation burden due to chemotherapies as adults and metastases.
So one of the other big themes here is global differences in genetics, host genetics. And, Mila, you commented on that with APOBECs, and Hannah also with respect to, immune alleles, MHC and HLA. Are those correlating at all with global incidences of cancer or outcomes, better or poor?
Hannah, do you want to give a simple answer to that?
I don't know if there is a simple answer.
Certainly seen examples of polymorphic variation that's associated with higher incidence of, for example, EGFR, exon 19 microdeletions, and I think in Asian ancestry backgrounds and some in South America as well. I do think there are ancestral differences. Definitely there's different frequencies of MHC alleles in different populations. But there's so many MHC alleles, though, that statistically it can be a little challenging to try to detect associations with particular mutations in particular groups.
Yeah.
I can add to that. This is just a very difficult question because we need to consider so many confounders, and that's why genetics is unlikely to answer this question alone. So we need to have functional data and also proof that the functionality of some of these variants we are considering, not just because they're statistically significantly associated, but actually shows that they are doing something different compared to many linked variants, which could be traveling along with the same haplotype.
Absolutely. So there were, hold on here, there are some questions appearing now too. One is a thanks to NCI for hosting this. Another is a general one of can the video and all of the presentations be released? I don't know the answer to that. And
there will be.
Okay, it will be.
Just to clarify, they will be released if people sign the consent for the presentations to be released.
Okay, thanks. And then can signatures be used to, this is more scientific, help with early cancer detection based on circulating tumor DNA? So just, if it's being screened in the blood, potentially, if there was some reason to do that.
Yeah, I would say potentially. I don't know exactly how, but yeah, if you could identify them the same way that they are informative in some cases, if you can identify them in the tumor, and in a similar way, they would, yeah.
Okay. Great, we're getting close to the end and our organizers are coming back. does anyone have another burning question or comment before we turn it over to our leadership?
No, just thanks, as someone in the audience said, thanks for organizing that. It's a great meeting, thanks.
Yeah, well done.
Okay, thank you all panelists for excellent talks and engaging discussion.